This is the end of the semester for E340, 599. The 15th week. Next week's the 16th week, the end of it. Appreciated you all being here. If you're interested, I'm teaching a class numbered 443, 543. That's on spatial modeling, multicellular spatial modeling next semester. It, in principle, is a 2-semester sequence, but in fact, there really isn't any prerequisite in the sense that they can be taken independently of each other, although it's certainly helpful to have had this material. You'd all be welcome if you wanted to take it. Same time, same structure, a little bit more developed because it's material that I've taught many times. But if you have any questions about it, please let me know. So today, the only topic that we have for class is the presentations. The order I had was Mercedes, Tyler, and Kali in that order, and then Josh, if we have time at the end. I know he'd like your feedback on his demo, and so he'd appreciate that. Any questions or discussion before we get started? No, everybody okay? Okay, then I'll turn it over to Mercedes. I'll unshare. So is it okay to record your presentation?
Yeah, that's fine.
Okay, great. And I'll mute myself until we come back to discuss.
Sounds good. So my name is Mercedes Olson, and I decided to do my project on modeling cancer tumor cell treatments using radiation and immunotherapy at the same time. So just to do a little background on cancer, I'm sure most of you have heard of it, but cancer is a genetic disease that is caused by changes to genes that control the way our cells function, most importantly, how they grow and they divide. The cancer cells differ from normal cells that they're less specialized, as in they don't mature into specific cell types with specific functions like normal cells do. And then they also ignore signals that usually tell the cells when to stop dividing and when to die, so apoptosis. And then another thing that you've probably heard of is also tumors, which are abnormal masses of tissue that form when cells grow and divide more than they should or not die when they should. And they can use the immune system to stay alive and grow. There are 2 different types. There's benign and malignant. And we're going to focus on malignant, which is a cancer tumor. And then just background on the treatments as well. There's many different types of treatments: radiation, immunotherapy, chemotherapy, several more. But this paper really focused on radiation, which is a treatment that uses high doses of radiation to kill cancer cells and shrink tumors. So same thing that's happening in X-rays. And basically what happens, the cancer cells whose DNA gets too damaged and cannot be repaired, they stop dividing and die. This takes days or weeks of treatment, depending on where and what the cancer is. And then after the treatment's done, the cancer cells continue to die for weeks or months afterwards. And then immunotherapy is a biological therapy that helps your immune system fight cancer. It uses substances made from living organisms. And again, depending on the type of cancer, the treatment can be every day, every month, every week. It can also be given in a period of treatments and then have an arrest period afterwards. And then both of those are really just cancer dependent, but they're what I chose based off of the paper for this project. And then the paper itself was called the optimal radiation dose to induce robust systematic anti-tumor immunity. And basically the main question was, what was the effect of radiation on the primary tumor, while also using the immune system through immunotherapy on the secondary tumor. And then their main goal seemed to me was to help get closer in finding the ideal amount of radiation. And then the summary of their aspect was the synergy of radiation and immune systems is getting a lot of attention because of all the potential benefits are shown. They are investigating the effects of the radiation and what that has on the immune system and strong anti-tumor responses. They use a math model to intentionally use radiation and immunotherapy on one tumor, which can also help irradiate a second tumor at a different site, which is with its own independent volume. And then they used published experimental data for this setup as well. So the biological model is that, like I said earlier, radiation alone is a treatment that can get rid of abnormal tissue, and it's a very common method. It can also induce stress of the immunogenic type of cell death, which causes the abscopal effect, which is a systematic regression of metastatic nodules outside the radiation field after a localized irradiation to one or more tumor sites. And studies have shown that it's immune mediated. And then in hand with that, immunotherapy also causes the activation of the tumor-specific cytotoxic lymphocytes, which attack that specific type of tumor. So basically in the biologic model, 2 tumors were grown in one mice at 2 separate sites, and then were then treated with radiation and then with and without immunotherapy. The adjusted amount of time for both, and then throughout the model, I adjusted the amount of time and amount of both radiation and immunotherapy to see the trends of the cell populations. I grabbed a schematic from the paper. I just thought it was kind of a nice visual to look at. And basically it shows that there are different types of cells in the circles. And then the arrows show the transitioning between compartments. And then where there are the little T-bars here indicates of the killing of cancer cells by the immune cells. They use the carcinoma cancer for this study in the paper. And then you can see that the one here is a little bit larger in the primary site, and then the second is a second tumor. Some of the mice in the study also received immunotherapy, which released the specific CTLs, or tumor-specific cytotoxic T cells. And then you can also see just where they flow as well. So moving on to the math model part of it. The main goal of the math model was to simulate the radiation response and induce the immune response, as well as the response from the immunotherapy dose. Again, there are 2 tumor sites with 2 separate locations and their own volumes. 4 of the main important parts were the viable cancer cells, which was denoted with C. And then there's that constant rate that they grow at, and then multiplying by both the total volume and the K, which is the tumor capacity, and then subtracting that alpha rate of its tumor viable cancer cells, and then E of I, which eventually is the immune response. Then they have the cancer dying cells in 2 ways, both the non-immunogenic manner and the immunogenic manner. And the non-immunogenic manner, which is denoted by D, you see that there's this rate here, again multiplied by the cancer in ratio to the total volume and the carrying capacity, and then added this time with that alpha rate. And I'll go over these variables again in the implementation aspect. And then there's the cancer cells dying of the immunogenic manner. So basically when the irradiation happens, this is when they die. And then there's a total measurable volume, taking into account the cancer cells and the 2 different dying types of cells. And then this is with really just the kind of the base of it. And then moving on, you add the radiation to it using these 3 equations. And that's just at that one radiation point. And then the final step would be to add the effect of the immunotherapy, which is this E equation multiplied by the lambda, which would be which is the rate at which the immune system returns back to its initial value. E star here is kind of more of an equilibrium point. As of right now in the model, I never adjusted that, so it could be elaborated on in the future. And then you continue on multiplying it by the rate at which L is times the E of T and then the volume. And then here is the U of T, which is the immunotherapy aspect, and then the W2, which again I'll talk about when I go through it all, but that's a T cell regulator, and then the 2 irradiation sites, and then the ratio of the volume here on the right. And then U of T, which is the aspect of the immunotherapy, was simplified. Was simplified from the paper just to keep it within the scope of the class. The paper originally had it with an integral, so it was just multiplied by the lifetime at which the immunotherapy will work, the 1 over it, and then multiplied it by again. And then, like I said, these 2 were adjusted from the original paper, from errors in their side, and then just simplicity on our side of the class. So going over the model implementation, The way I went about it was I first just made a singular tumor using those first 4 from that math model slide, the viable cancer cells, the 2 different dying cells, and then the total measure. I then replicated that exact same model, but added a second tumor, focusing really only on the radiation effects. And then both the adjusting the radiation on the time and the amount. And then the final thing I did was I added immunotherapy to the tumor sites by adding the components and incorporated the correct effector cell equation for the cytotoxic T cells. And then here's that chart I was talking to you about, just kind of going over the different parameters. That rate R was the viable cancer cell doubling time with the unit of 1 day over time. K is the tumor carrying capacity, which kind of sets the limit. A is the CTL's killing rate site, which is kind of why you only saw it when there was the immunotherapy aspect involved, because you need that for that to happen. Then you have D, the clearance rate of dying cells, which is another rate of 1 day over time. The SFD here is what was adjusted for the amount of radiation dosage. And then we'll mess with that a little bit more when I get to the notebook. L here is the decay rate of effector cells. So again, kind of why you only saw it when the effector cell equation was taken into place. W is the baseline T cell recruitment rate. W2, which we'll again look at in the sliders on the NanoHub application or notebook, shows the baseline of the T cell recruitment rates due to the immunogenic cell death. And then E is the folding rate change, and then CLR is the immunotherapy clearance rate. So I'm gonna stop sharing this presentation here for a second and then open up my NanoHub notebook. I don't quite have it ready 100% for the application. I thought it'd be more important to kind of get the presentation stuff out on the presentation for you guys to see. So there'll be some more meat and density to the, I just wanted to get.
Fine. That's fine. You have more time to finish it up.
Yeah, I just wanted
Thank
to
you
make
for
sure.
being willing. Thank you for being willing to be the first presenter. That's always takes some.
I'm definitely nervous, don't worry. But yeah, so I just really wanted to get the bulk of the model running. So it's missing some of the things as well. So can you guys see the notebook?
Yep.
Okay. So here's just that diagram again, and I've been over all those things in the presentation. So going into the model. So here I ran just that singular model, and then kind of seeing it in the NanoHub application, you can kind of see where the equation was changed to the tumor growth rate, the tumor clearance by immune cells, and just the competition for resources and the clearance of dead cells. Here's the the T cells. This isn't as relevant in the first part of this because I haven't added the immunotherapy as well, but then there's the decay and radiation. Here's when the first tumor happens, both with the total volume, so you can use it in the equations above, and then the dying immunogenically. And then it happens at T1 where the radiation is taken place. So here are those parameters again. Again, most of the values are kind of based off of the paper that was used, but then I chose to do the radiation, the amount of radiation as 0.2 for a base, and then the time that the radiation took place is at 60 days. So and then just in this first one, I did some super basic initial conditions, and then you can see here that the cancer cells come up, the cells are dying just because there's more cancer cells, so there's competition for resources, so they obviously die. Again, E is pretty irrelevant here, mostly because there's no immunotherapy, but right as the radiation hits, you can see that there's a spike in the cell death, and then it goes down for a little bit as the radiation is still taking place, and then it goes back up. So that's the super basic visual of how the model works. And then here, we're moving on to the second model, which is more based off of the paper, but still not really bringing in that immunotherapy aspect yet. This is just bringing in 2 tumors. Again, very similar, just replicating it. Here I made the first tumor a little bit larger with the initial amount of primary cells, just to kind of keep it consistent with what the paper saw. And then here it's changing. This first one, it's just the same exact thing that you saw as the second one, just with the 2 tumors and the two respective values based off the amount of cells that originally started. And then in the second graph, you can see the W2 is changed to 0 to just kind of represent how there was no change in the baseline T cell recruitment. So it's just something to look at. And then later on, we'll adjust what W2 looks like just in one tumor. So first thing I did was adjusted the time of radiation within a couple of the, within the tumor, both in tumor 1 and tumor 2. So I left the base one up and then I added 2 of the sliders. And basically the only reason that there's 2 sliders is so you can more clearly see both the primary and the secondary tumor and how they're reacting. So if you go to below the original days, both 34, around 34. Let's see if it'll run. Should be running. We might have to restart the notebook. This has been happening to me all day. Sorry about it.
That's okay. It's a friendly audience.
Yeah. I'll just kind of exit out of it. Page, and then we'll just reopen it. All right, here we go. Going over it again. So back to adjusting the time of the radiation. So let's see if it goes ahead and works this time. Yeah, so if you change it to 30 and 30, sometimes it's kind of hard to get it right on top of it, but basically you can see that there's really just not enough time for the cancer cell to even produce. So you hit the radiation, but then it just comes right back. In the second tumor, you see it just with a little bit lower, just because it's a little bit smaller tumor. And then if you were to like increase it to greater than the 60 days, you can see the results of what goes on. And I have screenshots at the end of the presentation to get a more overall view of it. So we can talk about it a little bit more then. Going on to adjusting the amount of the radiation, which I found a little bit more interesting, was so in the first tumor, and again, same thing with this one, I just have it in 2 separate ones. So it's not, you're not looking at 10 lines all overlapping each other. So the original number was 0.2. If you go to a little bit less than that, you can just see there's a smaller effect. And then as you increase the radiation, more of the cells, you see that spike in the cell death due to the immunogenic. So there it is again, just kind of spiking up as you increase it with more radiation, which makes sense. The more radiation you hit, the more cell death you should see. And there's a little bit longer of a lag in bringing the cells back, just because there's more hit with it. So then this one, I allowed for you to mess with both the radiation amount, so just kind of hitting it back with that too, and then the time of radiation, moving it to 60. So you can definitely see, and you can like adjust it. So what happens when you do it more radiation, and then you lower the time it happens, and then as there's more radiation, the spike in I is going to increase every time, and then the longer you bring it out, there's more cancer cells to kill. So then therefore there's more radiation, or the more radiation you add, the more cells it will take out. And then So the next part I did was adjusting the baseline T recruitment from the immunogenic cell death, which is that W2. And then this was just something so you could see just one of the parameters changed. So then just simple, and you can just see that as you increase the W2, and I believe it was 15 something in the paper, it takes longer for the cancer cells to come back because there's more T cells fighting it. So now going in to adding the immunotherapy, run that. Now you can see there's this huge spike where the immunotherapy happens, because at the same time the radiation is happening. But now you've added lambda, which is how long the immune response reaction will last. You've added E star, which is an equilibrium point for the immune response to go back to normal. I left that at 0. In the future, that's a parameter that could be changed, and seeing how your immune system is responding. And then lifetime is just set at 10 for the time of days for the immune response to kind of settle back down. Again, I left these values the same, but here is the immunotherapy component coming back. And then here are both at the first tumor and the second tumor, the cytotoxic T cells from the immune therapy effects coming into play.
Mercedes, I have a question.
Yeah.
How does the immunotherapy interact with the other variables of the model? I guess the other question is, the other way of saying it is, how is the immunotherapy represented in the model? What is the effect of adding immunotherapy?
It takes longer for the cancer cells to come back, so there's more death in the cells. So it just makes the treatment more efficient. And it's kind of fun to see how long. Well, I'll show you. Moving a little bit forward, but so then I ran it here with the lambda of 1. So then there's that huge, and then lambda again is the rate at which the immune system returns back to the initial value. So the bigger the lambda, the smaller of the immune effects, and then the smaller the lambda, the bigger the immune response is. And it's basically just kickstarting the immune system out of regulation and into high gear to fight off and bring the effector cells. So you no longer are you only just having the radiation cells, you now have the effector T cells, both fighting the cancer, which I guess answers your question a little bit, that it now brings in more cells that are fighting the cancer. And then this is just 2 visuals to kind of see what happens when there's a low lambda. You see right away that there's that huge spike, and then almost the whole entire tumor comes down and dies. It does again with the lower lambda as well, or the higher lambda. When you adjust the immunotherapy, the amount of lambda, so here you can mess with the amount that you want to do, so the time it takes. So I left the normal one up, and you can see when you have the lambda, which was set at, let's just go back up so I don't tell you the wrong number, set at 20, which is, and it's 1/days, so that's the unit for lambda. Here's that it goes away, and then with the you can see that the time in days is now longer. It's at 200, whereas just from the radiation I just had it at 100, and you could already see the cells coming back up over here. Now that you've put the immunotherapy into place, it's taking much longer for the cancer cells to come back. And similar with up in the radiation, here there's again just two graphs, so you can see just the reaction from the tumors easier without looking at 10 lines. So changing it to below the 20. Oh, I don't know why I did that. But you can see that when it's that higher, when it's that smaller number, there's that bigger immune response, and therefore it irradiates the cancer altogether. I'm not sure how accurate this would be to real life. It doesn't work like that. So that's one small law in the model, but you can just see it. And then if you increased it up to like 30, again, now it's a smaller immune response, and the rate's going back. So the cancer cells start reappearing at an earlier time. So now you can change the time of the immunotherapy with keeping it at that 20. And just see what's going on here. So basically, you can change it for, so it's the time of radiation is set at 60 days, and you can change it for before radiation. And you can see that there's almost no effect when you change it before the radiation hits. The immunotherapy wasn't as effective until you put it with the radiation. And then if you did it later than the radiation, you can see that when it happens here at day 75, there's a small indent. But again, right where the effector cells start to come into place. But it goes right back down and cancer comes right back up. So you can see that it was most efficient when it was at the same time as the radiation. As you can see, the tumor cells don't come back quite as fast, or the cancer cells don't come back quite as fast. And then again, this is just the second tumor, which will give you the similar results, just a little bit lower values. And then something I did to kind of wrap it up was allow you to change both the tumor radiation time and the immunotherapy time to get the, excuse me, to just kind of see the relation between it. So again, here's the baseline of 60 days what happens, but what happens when radiation is at 45 and then you put the immunotherapy before it, or you put the immunotherapy after it, and then vice versa as well. So, and then this will be added on to get ready for the NanoHub app. But that kind of sums up all the different parameters I went on to. And then I'll kind of sum up the results from the observations. Going back to my PowerPoint here. All right. So I spent a little more time back in the notebook about the radiation effects, so I won't spend as much time again here. But you can just see the sooner you hit it with radiation, the less cancer cells it can get, and then the cancer cells just come right on back. Again, you can kind of ignore the E equation in this because it's not as accurate as when you get the immunotherapy, and this is really just focusing on the results from the radiation, so there are no huge effector cells playing a role here. And then if you make radiation last a little bit longer, it spikes up, kills the cancer, and then it comes back. It seems that the most ideal day when running through it was at 60. And then you can change the amount of radiation, like I said. When it was at smaller level of radiation, there wasn't as many cells killed, and it came back right away. It doesn't even look like they all disappeared the way they do with a couple of the other simulations ran when you add the immunotherapy. And then when you add more radiation, it spikes up here with the cell death immunogenically, and then it comes back down. And as you can see, when you look at the 100 value here between the time with the higher radiation level, there's less cells, so it worked a little bit better, which seems pretty intuitive when you think about how much radiation you're hitting at the tumor site. The immunotherapy results, again, here's that baseline one, hitting the immunotherapy with the 20 for the lambda rate, and then just looking at it base-wise. The top is showing you adjusting the time. So again, when you hit it before the radiation, it does little to nothing. You can see the little dip here at 40, where the effector cells are coming in and trying to kill some of the tumor, but it was most efficient when you hit it with the tumor, as you can see, because there's no cell deaths coming back around 100 like there is up here. And then when you hit it after, again, still not as effective as when you hit it at the same time. You can see the little dip again at 80 and the little change, but it's not really stopping it the way the radiation had this dip here. And then when you change the amount of radiation or immunotherapy, the smaller the lambda, the bigger the immune response is from the T cells. And you can see that it really did a good job of eliminating the cancer cells altogether. And then the larger lambda, like I said previously, has a smaller immune response, and then around 150 days, the cancer starts coming back. Excuse me. And then the both results, just to summarize, I know I didn't mess with the toggles a lot. I thought that this would just be easier. When both treatments happened at the same time, it takes a longer amount of days for cancer cells to start increasing again, which is this top graph here. When the immunotherapy happens before radiation, there's really the very small effect from the effector cells that have now been released due to the immunotherapy. And then it again goes down when you hit that radiation, but it goes right back up as they're not doing that synergy. They're not doing that working together thing. That's really spiking in the popularity right now with the research. When you do the radiation before the immunotherapy, you get that same effect. You get that effect from the radiation. You hit the immunotherapy, and it's got that little tiny dip, but then it just stays right up there. And then, so I kind of, if I were to continue this, I'd want to look at what happens when you do multiple moments of radiation treatments along with multiple moments of immunotherapy. I'd look more ideally into the specific amount of the radiation and immunotherapy, what one was the best. I know the paper made a conclusion on it to help for previous research. I didn't look enough into it with the model to be able to make a conclusive number the way they did. So I think it'd be really interesting to see how much radiation. And then a long way down, I'd be interested in what type of cancer does this change? Does it act a little bit differently? Does is it if it's a life-threatening cancer, or if it's just a skin cancer that can be treated? So if that has anything to change with it, that's kind of what I'd take this next step in. And then obviously the notebook has a little bit more work to be done before it's an application. But this is the source of the paper. And then I used this website several times for the background information for cancer. So that is where I'm at for the final project.
Okay, thank you. Good job. Comments from the questions, suggestions? As I say, you could please write them down, but also we could have a discussion. We didn't really talk about cancer much in this class, but it's an important topic. It's interesting because I heard a talk this afternoon by Bart Ehrmantraut about a response to COVID infection, and the model structure was actually very similar to the one that you have in this model. But before I say anything, I should want to let everybody have a chance to contribute. Anyone have particular questions or comments they'd like to make? Don't be shy, please.
Did the paper give any like threshold for eradication?
Like how, like what the different, I can't think of, like GYs they used. Is that what you're asking about? Like the levels they used?
Yeah, kind of like at what point can you say that the cancer has been eradicated, that it's been treated, and it's now gone?
That's actually a pretty great question. I know, kind of going back to here, these are the doses they used.
Right.
But I can't answer that.
Okay, I was just curious.
Yeah, no, I understand. I would assume it's when the tumor is no longer like there, is when they were using it, is when it's been totally eradicated, is when both, not only the primary tumor, but also the secondary tumor was gone. Sorry.
Right. Okay, cool. Thank you. I think your code and your project look great.
Thank you.
Was it Brian? Did you have a question?
I just said it was well presented. I think the template was really nice.
Thank you.
I guess anybody else? I had one or two comments, but nothing dramatic. I think if we come back to that first picture that you have, the one from the paper, and maybe there's a question, a little bit of the model, what you modeled. Might be nice if you could explain a little bit about what immunotherapy is and does. and radiation is and does. I know when you're working on writing the code, then writing everything up is, you have to do a lot of things at once. But if you look at this picture is maybe slightly different from what you were explaining in the simulations.
Yeah.
And the big thing here is that typically with radiation, you can only irradiate the primary tumor because you don't know where the secondary tumors are.
Mhm.
And so I think that the key question here, perhaps, at least in Heiko's paper, is if you irradiate the primary tumor, you maybe could kill all the tumor cells of the primary tumor. But if you're not irradiating the secondary tumor, then you're going to die from the secondary tumor. And so the question is whether the immune response caused by killing the primary tumor is sufficient to kill the secondary tumor, even though you don't irradiate it. And so you gave us a nice example where you irradiate the primary tumor and then you irradiate the secondary tumor as well. And that's interesting. But I think one of the key things that's interesting is what happens if you only irradiate one tumor, and what happens to the second one. And so you could show the time series. What I might think about doing would be first show the time series if you don't do any radiation or immunotherapy. Both tumors grow to their carrying capacity, and that's it. That's your reference. And now if I irradiate the first tumor and there's no immune response, the first tumor shrinks. Maybe it comes back later, maybe it doesn't. But the second tumor keeps growing because it doesn't see the radiation. Then the third thing is now I add the immune response. I irradiate the first tumor, it shrinks. The immune cells come in and they kill both cells in the first tumor and the second tumor. So the second tumor shrinks because of the immune response, even though I didn't irradiate it. And now if I apply immunotherapy, the second tumor shrinks more because I've amplified the immune killing of the second tumor. And so I think the outline that you followed is very good. But maybe, and the questions you've asked are fine, but maybe it would be worth thinking about those things a little bit and maybe pursue them a little bit. Because I think that there were a couple of times, I think, when you said radiation where you maybe meant immune killing or vice versa. And so it would be worth checking that because it's pretty interesting that you're able to eliminate with the immunotherapy, you're able to eliminate the tumor. But I think the key question is, can you use the first tumor to get at the second tumor fundamentally? Can you use the tumor you could see, which is the one you could irradiate, the primary, to get at the tumors you can't see, which are the secondaries? And so then maybe we need to look at the equations to make sure that the immune cells are coming in and actually killing the secondary tumor. And so we could come back. We don't have to do that here, but we could meet tomorrow or later in the week and just double check to make sure all those equations are actually doing the thing you want, which is the radiation kills the first tumor, but with no immune response, it should do nothing to the second tumor. When you add the immune response, you kill the first tumor, you see the second tumor shrink. You now provide the immunotherapy, and it was very interesting to see the times courses that you did where you changed the time and amplitude of the immunotherapy. But that should have an effect both on the first tumor because it cleans up what didn't get killed by the radiation, but also on the second tumor.
Okay.
And so the stronger the immunotherapy is, the more the second tumor shrinks when you irradiate the first tumor. And I think all the pieces are there. Maybe we could meet later in the week and try to do that. But I thought I was also very impressed with the presentation. As I said, maybe a little bit more about what the immune system is doing, what immunotherapy is doing as a biological background.
Okay.
Then explaining where the various terms in your model come from, And that's a good exercise, because if you could explain them to your audience, then you're pretty sure they're right. And if you can't explain them to your audience, then it's possible you may be right, but it's possible that there is a mistake. And I know there seemed to have been a mistake in the paper, at least something very confusing in the paper. So it's possible that the model itself that you were working from had a bug in it, which doesn't help things. But conceptually, I think Josh actually pointed to one of the equations when you were talking to it, which was the immune response equation.
Mhm.
And I think the key thing is that there's an immune response that's triggered by the number of dead cells. And those immune cells get allocated to both tumors, not just to the tumor that caused it. And we should probably come back. We don't have to do that in front of everybody. And make sure that the allocation of the immune cells to the two tumors is what you want. My guess is that there are probably a lot of different ways one could do that, but it could be interesting. But I think the idea of the flow that you have of gradually adding complexity to the model in the notebook is great, and I think that the material you presented is very nice. So it's a good. Thank you again for being willing to go first, and jump in on that, and especially on a topic that's a little bit. In most semesters, cancer modeling would have been a main part of the course. Next semester, we'll probably do more cancer modeling, and so it was great to see somebody reach out like you did outside of the main topic of the course and try something like that. Other questions or comments? Josh, do you have anything you want to go over?
I have a couple of comments, but I'm gonna send them to Mercedes directly. It's just a matter of how to illustrate in the app what the different models are, because you should you should walk the user preferably with diagrams. When you say model 1 that has no secondary tumor, then you have just a diagram of that, what the cells are doing in that secondary tumor.
Okay.
And then primary tumor. And then when you go to add more complexity, that you take that diagram and extend it. And I can walk you, like I use PowerPoint to do those diagrams. I don't use anything fancy or anything.
Okay.
And you just have to copy it from whatever they did in the paper. But that helps me out because I knew what you were talking about, but I was thinking of a user that has to walk through the app, and then you say, okay, now secondary tumor, but that, like, it's better to illustrate what the added complexity is doing in terms of a flow diagram.
Yeah, especially for somebody who's never seen it.
Yeah. And then I have the question. I actually have the question of what is the relation between the primary tumor side and the secondary tumor side? Like, do you have cancer cells going from one to the other, or is just the way they elicit the immune response?
I don't think I have them going back and forth to each other.
Okay.
I
so
think
it's
that
just
independent.
Their only interaction is that they share the immune response.
Yeah.
Okay.
Correct.
Okay, cool. So that's important to explain.
Okay.
So yeah.
And as I say, we just heard a talk today by Bart Ehrmantraut, who was modeling COVID. And in his model, there's an infection in the throat and an infection in the lungs. And the immune system responds to both rather similarly to what you have here. It's not quite the same, but it's the kind of situation also where the immune response, the one of the regions triggers an immune response that then affects the other region. And that's pretty typical of the immune system, because the immune system responds to everywhere in your body, but the signal that's triggering it may start only in one place. That's one of the reasons the immune system is powerful. It also can cause problems in some cases. So let's plan on meeting up and going over the details and helping you find that. But that's really great. And the big annoyance was that it winds up that LT is a reserved word in antibody. So don't call a variable LT. Took Andy to find that one. But I appreciate that. Despite that, you were still willing to talk today. So I appreciate that very much. Okay. So Tyler, are you ready to give us a presentation? Is it okay if we record it?
Yeah, that's totally fine.
Okay.
Okay. So my name is Tyler Hanna, and my presentation is over the in-host viral kinetics of influenza A. So some biological background is that obviously we all know what influenza A is. It's an annual epidemic that we deal with every year, and the peak infection rates are what we know as flu season, and that occurs in the fall and the winter. And the reason why we need a new vaccine every year is that it mutates every year due to antigenic shift, which is where the virus picks up minor mutations that make it just different enough that our body can't recognize it, and we need a new vaccine. And the big issue with this vaccine is that it's not all that effective, it seems, for everybody. So, and especially since not everybody gets it every year, it just seems to never go away. And although it seems to always be around, we still don't understand the kinetics of it during an infection. And especially with the immune response and the specific mechanisms seem pretty unclear regarding the adaptive immune response and the memory. And have antiviral treatments for it, such as Theraflu. And the problem with these treatments is that although they are really effective, you need to take them at the very start of the infection or prior to, which is really hard to determine. And like I said before, due to this unpredictable mutation nature of the virus, it's really hard for us to see an end to it. And the thought of a universal vaccine is still being researched. There still needs a lot more research to be done on it. But also, yeah, I mentioned the current vaccine we have is not too incredibly effective, as it only works for about a year. And the purpose of this paper really is to better understand the kinetics of it, and in combination with our immune response we have, so that we could possibly look toward a better treatment, a new type of treatment, or improve upon our existing antiviral treatments, maybe to better recognize an infection before it gets beyond the point of antiviral treatment effectivity. And the main paper I used used a fairly simple model of infection. It's basically a very simple SI model and it builds on complexity throughout to consider our immune response. But the limitation of this model is that it's mainly stuck in the upper respiratory tract. So the virus could behave differently if it were in the sinuses or in the lower respiratory tract, and so on. And with the actual viral load, it does show a bimodal double peak behavior, and this is explained due to the incorporation of interferon as the model gets more complex. But it also could be the result of the virus spreading to a different respiratory tract, where there is a more abundant source of target cells, which could lead to a spike in viral load. And in the original paper, it did include an immune response. However, it was very implicit, as it only described it as a general death rate of infected cells. However, we know that this is obviously not the case. So I consulted a second source that very specific on the immune response, and I pulled certain elements that aligned with the model I was currently using in order to better define this death rate and exactly how it works. So as I mentioned before, It's just a basic SI model to start out with. Starts out with an initial amount of susceptible target cells and an initial viral load. And since there is no recovery factor of the cells, typically the infection will result in all target cells being infected, or if the infection isn't very efficient, then there will be some target cells left over. The more complex model considers the delayed interferon production, and interferon is produced from infected cells, and interferon is not just produced from infected cells, but for the purpose of this model, it was assumed to just be from infected cells. But interferon is in fact secreted from monocytes and macrophages as well. And here is the and equations for the first model. As you can see, it's just a very simple SI model. Here we have this beta TV term is the rate of infection from susceptible to infected for the cells. And this delta is the general term for the death rate, and also acts as the implicit immune response. This parameter P is the viral production from infected cells, and C represents the rate at which free virus decays naturally. And here is where the model builds a little bit more complexity, adding the interferon effects. So now it adds a different category of infected, where there is non-productive infected cells and productively infected cells, which is I1 and I2 respectively. And this factor K represents the rate of transition from non-productively infected to productively infected. And the presence of interferon is thought to affect this rate. And as you can see, this second to last equation down here, this epsilon term multiplied by the amount of interferon in the system at that time will decrease K, which will decrease the transition rate. The same thing happens to P, which is the viral production rate. As P decreases, the amount of viral load decreases, which basically sums up the purpose of interferon in the system. And this S times I2 times 1 minus tau term represents the delay of interferon production. And that term I will explain in the notebook later, but it's more of a complex, it's pretty complicated to replicate in the model. And I was able to get some results that I will show you later. And then we still have our delta here. That is the death rate of infected, productively infected cells. And alpha is our term for the degradation of interferon in the system. And the degradation this alpha term summarizes both natural decay of interferon as well as interferon that is used up, because interferon will attach to receptors on the cell and make them more, I guess, resistant to becoming infected. And then to extend on this model, to better define that delta term, I consulted the second source, which described the use of effector cells, which are produced by antigen presenting cells. Excuse me. There is also another assumption made here where effector cells were only produced by antigen presenting cells. But this is also not true, as they are produced by others. But for the purposes of this model, we just kept it to infected cells. Here, APC is known to produce effector cells in response to the presence of infected cells or free virus in the system. And as I just mentioned, in this model, it's assumed that only infected cells trigger this production of effector cells. And according to this error diagram, we still have our same transition rate from susceptible to infected. And however, we do not have an I2 term anymore, as this model is an extension of the original SI model, but just a better defined delta term. And here, this m times I, this m term represents the rate constant at which effector cells are produced in the presence of infected cells. Infected cells still produce free virus at a rate p, and virus still has a natural clearance rate. Where this used to be delta times I, we have a natural death rate as well as a death rate associated with the activity of effector cells. And as I'll explain later, effector cells have a limit to the number of infected cells they can kill in their lifetime, which when they kill that number of infected cells, they will be used up or die. And they also have an associated natural death rate. And as I explained, in the model with delay is where the mathematics get a little complicated. With the redefinition of the transition rate as well as the viral production, we have this redefinition for K and for P, and these represent the lowering in the rate of transition and the lowering in the rate of viral production from infected cells due to the presence of interferon. And furthermore, there is another model which considers treatment with Xandinor. Oh, I should know that. I forgot the name of the treatment, but it is an antiviral treatment that will further decrease this parameter P, and thus it's an inhibitor. It inhibits an infected cell's ability to produce virus, which will in turn shorten the length of the infection. And now I will go to the notebook. So here we have the viral data for each. Well, first I'll say the paper includes data from 6 individual patients, and here we have the viral titer for each of the 6 patients. For the purpose of fitting parameters and finding the best fit model for this data, we use the average data, which is shown here. And here I'm going to demonstrate how adjusting the parameters will change the overall outlook. So first I'll start with P, and by changing the value of P from an initial value of 0.5 to 0.1, we see that the course of the infection seems to be delayed by a day, and the viral peak will not only reach a peak, but the peak will come later in time. And now if we change beta, beta is very sensitive, so it's hard to get a specific value. So I'll just show by gradually increasing beta how the infection will behave. And beta represents the infectivity of the virus, pretty much the probability that one virion is going to infect one cell. And as this parameter is increased, each virion becomes more and more infective, which is going to spike the rate of infection very early on. It doesn't necessarily increase the viral peak, as there is a limited number of target cells. So there can only be so much virus in the cell really at one time. I reset that. By adjusting delta, the death rate of the virus, we will see how that affects the rate of the infection. If I set it to 10, you see that the overall, like the peak of the virus comes later, as well as the maximum value of that peak will decrease. And the opposite is true when delta is decreased.
So I'm just quick suggestion here. Try to fix the axis of both of your plots. One of them is gonna be fixed always, the one on the left side. Yeah. But the right side, if you want to get an idea of whether it's going up or down, like a better, because the axis is readjusting automatically.
Yeah.
Try to find ranges that are reasonable. and then fix those ranges. So as you change the slider, then you will actually see the peak going down instead of the axis readjusting.
Okay. Do you mean
So yeah, like now
Yep.
when you change what was the variable?
delta?
Delta. Yeah. As you change delta, the peak is moving too. It's not just happening earlier, it's also decreasing or increasing, right?
Yeah.
But it's hard to see because the right side readjusts it's readjusted automatically. Just move it for a little bit. So you see how it's readjusting.
Yeah.
So you want to fix that. You want to fix it so it's much more apparent what is happening with the peak.
So don't auto scale
Right.
the logarithmic plot. Fix the
Right.
limits, the y limits on.
I can, and I'll talk to you tomorrow about how to do it. It's not, you just have to find the range where the maximum occurs.
Okay.
The other thing you might want to do is actually allow the user to change the duration of the simulation, so that because sometimes everything is over at 8 days. Sometimes you want to zoom in on the early time. For this one, maybe 8 days is enough. But for example, for Mercedes simulation, the user might want to be able to simulate for 6 months or a year or 2 years or 3 years. And so you might want to have a slider that controls how long the simulation runs for.
Okay.
Keep going.
Okay. So now we see how delta affects the results. Now, if we change the viral clearance rate, if we increase the rate at which free virus is cleared, we see that the maximum value on the axis here is dropping as C increases, and the opposite when we decrease C. Yeah. There we go. And when C is negative, well, according to the calculation of R naught, it'll give an indefinite value. But that's the behavior of the viral clearance rate in relation to the infection. And here we can mess with the value of R naught, which I believe the average value for this, according to this paper, was 11, which represents about one cell infecting about 11 other cells on average. And here, by increasing R naught, we see the infection spreads very rapidly in the start. And based on the data we have here, I've found that any value of R naught pretty much past 50 is going to show relatively similar results, as the infection only spreads like this. The infection at this point is already spreading so fast, it doesn't really make a massive difference what the value is. However, when R naught is around 2 or 3, we will see a dramatic change in the graph if it loads.
You have to wait a minute. It's still catching up with you.
I think I need to add a continuous update change.
Yeah, whenever you have a little bit of time, just go over each of your, and this goes for everyone as well. If it's taking a long time to update as you switch your parameter, you can change the continuous update to false. So then the model doesn't have to be updated every step in between the beginning and the end point.
It only updates when you let go of the slider.
Right.
And then it's faster.
So as R naught is now dropping, we see that the graph is shifting greater and greater with each step in the decline. Yeah, so
Here you see the maximum amount of virus going down, right? It's 10 to the 5th instead of 10 to the 7th or 10 to the 8th.
right. If I actually punch in a value, it might work better. there. Now R naught value is around 1. The viral load here peaks just over 10 or 20, which is basically nothing. And we see that the target cell count drops minimally. And here we have patient-specific data, which I should have included the table for the values. But here we have each model loaded for each specific patient. Patient, I believe, 1 through 3 showed the 2 peaks of the viral load, which, due to the fitting algorithm that the paper used, is very hard to replicate. But am I cutting out? Can you hear me?
You're all good.
Fine. I'm not sure what you mean by 2 peaks of viral load. In the patient data, I didn't see 2 peaks, but maybe they're.
For the model with interferon response, which I'll get to just in a minute. Here I have the fitting algorithm to fit the average data I mentioned before, and then we have a model that will run with the optimized parameters, which hopefully doesn't take too long. But while that runs, I'll explain the model with the interferon response. So this, is, like I said before, is an extension of the simpler model we just went through, where interferon is going to not only interfere with the transition rate from non-productive to productively infected, but will also influence the rate of viral production from infected cells. So I can't mess with the sliders until this is done.
Josh, we probably should have figure out how to have a stop fit button.
We were working on it. Oh yeah, that's another option. We can do that. Well, but the problem is that will require running the optimization in the background, as Andy was saying, because we can add a button that says passes through to the optimizer, so it stops. But the kernel will not read that until the optimization is over, unless the optimization is running in the background.
Well, the optimizer is in a loop, so I wonder if one could put inside of the function inside of the optimizer something that looks for a signal that does a break.
Right, like maybe if we put the button inside, we can try that. In the worst case scenario, we can try what Andy said last time.
But so it's still.
Yeah, this is taking longer than I expected it would do.
That's okay.
What as a proxy for that, what Tyler and I were talking is we have an idea of what an acceptable range of the error is, and then we can put that in the optimizer. So at least from a pedagogical perspective for the app, then it would stop much earlier.
Yeah, because here it's pretty much fitted.
Yeah.
But
yeah, so you basically know that 1.7, now it's optimizing up to more than 2 decimal places in the error.
Is it still running or is it done? I can't tell.
When it's done, it should spit out this optimized model. I thought I changed the like the minimum value it should be looking for, but I guess I didn't save it.
If it's taking too long, then you can probably go back to the code and
Yeah, this is getting a little much. Okay. Where I left off, here we're at the model with the delayed interferon production. And like I said before, to reiterate, the assumption made is that interferon is produced the interferon is produced from I2 infected cells, the productively infected. However, it is also produced from monocytes, macrophages, and plasmacytoid dendritic cells. But for the purpose of the model, we just assume that it's the infected cells. So to adjust the rate of P to be 0.1, we will decrease the viral production rate. And as we see the maximum peak of the viral curve is much less than it was originally and occurs much later, as well as the when all of the target cells are infected, it becomes much later as well. And as we see here, it doesn't reach 0, so we still have some left over. If we were to increase P by more, we will see the graph shift to the left and the viral load reaching a higher peak much sooner and a rapid plummet in target cells that will eventually reach 0. Really? Okay, the app keeps breaking. I'll just do it from the actual notebook. Okay, and here, now we're going to increase beta, and we'll see that an increase in beta is going to result in a rapid spread of the infection like we saw in the previous model. Yep, and the graph is going to behave similar to P. However, we'll notice that this interferon is also going to increase much more rapidly as the level of I2 cells peaks, which will result in a greater rate of interferon production. And by increasing delta between 0 and 20 is going to result in, so make it 0.2. We see that by decreasing the death rate, we're going to see that the infected cells are going to hang around much longer, which are going to produce more interferon as well as the viral load. So as you see, both of these end on very high values. And if we increase delta, we'll see both of these reach not only lower values, but they will reach their maximums later in the infection. And it will take longer for the target cells to all be infected as well. The parameter C, our clearance rate. By increasing the clearance rate, we'll see similar results. The infection is going to be prolonged, and the viral load is going to reach a lower value, as well as the interferon value. And decreasing will do the opposite. And by increasing our transition rate, we will see an increase in both interferon production as well as an increase in load, as the rate of from cells going from non-productive state to a productively infected state is going to increase the viral load as well as the interferon presence. And alpha here is our rate constant for the degradation of our interferon. And if we were to, over here I have sets of parameters that are closely related. If I were to D alpha, which is so lower the rate at which interferon is removed from the system and increase the rate at which cells become productively infected, then we'll see a spike in interferon production and a spike in viral load. And this could be thought to simulate like almost like a hyperactive immune system, and doing the opposite would decrease both the values, would decrease the values of interferon and also delay the infection. And tau is where the model gets a little tricky. So tau represents the delay of interferon production. So it's initially set to 1/2 day. So the rate of interferon is going to be based on the amount of infected cells there were 12 hours prior. And by increasing this rate, we will see that interferon will be produced. If I make it 1, then it will be interferon based on the presence of infected cells 0 days actually, or excuse me, 1 day later, which would thus result in a higher interferon rate, because if interferon is delayed, if the production is delayed, and that gives the infected cells a chance to become more abundant, and the more abundant infected cells are, thus the more abundance of interferon we'll see in the system. And the opposite is true if tau is decreased. And here I have individual patient data for each patient in the delay model, which is where I expected to see the double peak in viral load, but I, for some reason, could not get it to work. Here we should expect to see the screen line have 2 bumps for patients 1, 2, and 3. However, due to their fitting algorithm that they use in the paper, and it was very complex, and according to the assumptions that they made, I was unable to replicate that curve. And here we have the fitting.
So did they give you the parameters that they used that got 2 peaks?
They did. However, they did mention in the paper that they, due to the overfitting of their parameters for the delay model, and also some of the parameters they said were available upon request. So I didn't have access to, I guess, the precise values that they used or the algorithm.
And I guess I have the, and also I have the question as to, because the bucket brigade is an approximation of the delay. I'm not sure how the fitting is different between, because you have the tau here being a little bit different from whatever they use to fit their model.
Yeah, I don't know. I mean, if they don't give the values if they don't give you the parameter values that give the result, it's awfully hard to check.
Yeah, that was a little bit of an inconvenience. And here is the fitting. I won't run it just because it'll take a while.
But I don't see 2. I mean, in that patient data set, there's only 1 peak, right?
Right. And we should see. Should I
pull
up?
Show a patient data set that has 2 peaks, just so we could see what it looks like
in the original paper?
Okay, either that or okay.
This is what I should.
So they believe that itty bitty dip, huh?
Yeah.
And I think
I'd say if
that's
you-
the whole reason they added the interferon model, to be able to like add to get the double peak.
But if you look at their fits, they're not a big. It's not a big dip.
No.
Yeah. So the reason they need the delay is to get that second peak. Yeah. Okay. But probably fitting it without that second peak is pretty good. I mean, you're basing that second peak on one data point. So how reliable that is the question.
Yeah. Let's see. Here, yeah, there's a fitting algorithm, and here, here is the antiviral treatment model. So this is an extension of the previous model with the interferon delay. However, it includes the treatment of Zinminifir. And so here they considered the early treatment to be at 1.2 days, which results in this viral curve. And then they considered delayed treatment at 2.08 days. So here we notice that the later the treatment is administered, then the higher peak, the viral load peak, excuse me, is much higher. And this is consistent to the treatment I mentioned in the background of influenza. The earlier the treatment is administered, then we see that you almost have no viral production. It's right about 0. And as you delay the treatment, we see that it becomes less and less and less effective. And also, a key thing to notice is to point out is that this 97% effective rate was an assumption made in the paper, and I found that to be a little possibly unreasonable. So I added a slider to change how effective the treatment really is. There we go.
Yeah, one thing you might think about doing would be to have basically a static picture in the background of what you'd get if you didn't have any treatment. So basically put next to this picture, which changes, a picture above it, a picture which just shows what the viral curve is without any treatment, so that the person running the simulation can see how it compares.
Okay.
I mean, of course, in your simulation, putting T to be very late is equivalent to that, or putting the effectiveness to 0 is equivalent to that. But basically take a snapshot of that and paste it in so that the person knows what to compare to. This is what you'd get if you didn't have treatment. This is what you get with treatment of different kinds. But that's a great demo. I like that.
Right. So as we see, if you administer a treatment too late, then I think around the 3rd day, it seems to not do anything. And the same is true, obviously, later in the infection, you might as well have just not taken any antiviral treatment at all. So really, it's only effective before the 2nd day, or to take before the infection, which, as I mentioned before, that's really hard to determine when you have it, because you may not be showing symptoms until 2, maybe 3 days after you have it. So that's one complication. And here we have an extension that it looks to better define the immune response, which was summed up by the parameter delta. And if it loads, there we go. So here we incorporate the use of infector cells, and infector cells are secreted or are produced by the stimulation of antigen presenting cells in the immune system, and antigen presenting cells will produce effector cells based on the presence of free virus or infected cells that they know that they can detect in the system. And here we introduce 5 new parameters. We have K1, K2, D, M, and N kill, where K1 represents the rate at which effector cells are able to kill infected cells, and K2 represents the rate at which effector cells die due to killing infected cells. And according to the paper that I pulled, the mechanism for effector cells can kill about 100 infected cells before dying. So the ratio of, so K2 is about 100 times smaller than K1. And the parameter D represents the natural death rate of the infected cells and effector cells per day. And I assumed that D would be the same for both of these, as it's a very small number to begin with, according to the other paper. Our parameter M here represents the rate of production of effector cells due to the presence of infected cells. And here our parameter nkill represents the number of cells that an effector cell can kill in its lifetime. And here we see that if we start off by adjusting the number of cells that an effector cell can kill and increase the length of the simulation, we see that by doubling the number of cells that it can kill, this value right here of the, this end value of the viral load, the infection is delayed because as infected cells are killed at a higher rate than the amount of effector cells, the rate at which effector cells are produced is going to go down, as well as the number of infected cells, as we see here. And C is going to behave as we've seen in the previous models, where by increasing C, we're going to see a longer infection duration, and decreasing will do just the opposite. And here, DR represents our parameter D, which is the natural death rate of effector and infected cells, which is by increasing the death rate, it's going to shorten the length of the infection. And by decreasing it, it's going to, by decreasing the death rate, notice it has little effect on the initial proliferation of virus and infected cells, but it mostly affects the ending steady state values of both of these. And that's about as far as the notebook goes.
My guess is that assumption on the death rates is probably not quite right. Okay. If you look at the first model in Perlson's models, you have a typical lifetime for the infected cells. I think for influenza it's typically 2 days.
Okay.
I don't know what the typical lifetime would be for the immune cells. It could be long, depending on the immune cell type. If the infected cells are being killed rapidly by the effector cells, it doesn't matter. But the typical lifetime of the infected cells might not be as long as 15 days. And they may not be the same. I don't know. That's something to maybe look up a little bit.
Okay.
It's not an unreasonable guess, but I don't know if that's correct. But that's very nice. You've explored a lot. So instead of my talking so much, maybe Josh, let's let the audience have a set of comments. Was that the end? You said it was where, or did you have more to present?
Pretty much I pretty much talked a lot about the results in the notebook. This pretty much sums up what I've discussed about how the model is changed due to changing parameters, and pretty much a further explanation. So here are the results from the very first model. Is pretty straightforward. the model was pretty much replicated perfectly. However, the delay is where I kind of got a few issues with the bimodal behavior of the viral load. And I mentioned here, due to their algorithm, lack of parameter accuracy and whatnot, I wasn't able to replicate that curve. And then the treatment was consistent with the fact that if you take antiviral medication at the very start or prior to an infection, then you're able to clear the infection very quickly. However, if you take it past a certain point, then it becomes essentially useless. And here, as we saw, the model with the better defined immune response showed intuitive results. However, there was a lot of assumptions made, and that I will try to further make it more accurate. And with if I were to expand this model, I would like to really see how not only to better define the immune response as a whole, including the innate immune response like coughing and what mucus could do to the viral load, but also how vaccination can work alongside the system, and see how that dictates the course of a flu infection. And additionally, I wanted to see if different strains of influenza would behave differently according to the model I have set up here. And that's about it.
Very nice, thank you. So that's impressive. Questions, comments, suggestions from the audience?
I have the same suggestion that I have for Mercedes. As you have the diagrams of the model in the presentation, it would be also useful to have them in the notebook, because diagrams are very illustrative for people, in the sense that they can see how the variables relate to each other. And then as you explain the parameters, if you can put the parameters in the arrows, then people can associate very quickly how the parameter is related to the variables.
Okay.
I'd say the same thing with your sliders. When you display the slider, if the text on the slider can explain what the parameter means and what the units are. I noticed that you display it at the bottom underneath, which is nice, but actually in the bar that creates the slider, the text that creates the slider, you could actually put in an annotation that says, this is the rate of infectivity in units of per day or something like that. And so then they don't have, the person who's sliding can see it in the slider. They don't have to look down, just the way your speedometer says miles per hour in the speedometer. You don't have to look separately for that. That's a small thing, but I think it could make it a little easier to see. I mean, one thing about the model is that the effector cells aren't delayed. You've delayed the interferon and you haven't delayed the effector cells. In reality, the interferon response is pretty fast and the effector cells are slow. I'm not saying you should redo everything. I think that's all right because you're based on models that do it that way. But the effector cell response is generally takes days for that to happen. The interferon response is pretty fast, in the sense of less than a day. And so it maybe it's worth, at least in your text, discussing a little bit about whether it's okay to keep it the way you did with those decisions, but you should maybe discuss a little bit about where those delays come from biologically and what they mean. But I think I didn't have too many other comments beyond that. Again, I might have some graphs that are fixed graphs in the text boxes that show what you're comparing to. so that the person who's running it can see how it's different. That's more of a pedagogical thing. And then maybe a slider to control the duration of the simulation so they can zoom in on early times or make it run longer if it's taking longer to run for the things to happen.
Okay.
Again, we can talk about that. Other comments, ideas?
I was just going to say that this presentation, it kind of reminds me how I kind of structure my own presentations. So I just thought that was an interesting comment.
Again, I think we can all learn from each other. And in this semester, everybody worked on their own, and usually their people team up. But that does, even if you work on your own, it's useful to see how people do things and learn from it. Okay, that was fine. Thank you. We had that presentation a week ago. Let's see, was it last week? No, 3 weeks ago, just the week before Thanksgiving, Alan Perlson gave a talk, and we recorded it. So if you're interested in hearing Alan actually talk about these models, we have a video of him giving a talk about it. And it's a short talk. It's only 15 minutes. It doesn't take too much time. So if you'd like to, it's on our YouTube channel. So if you can't find it, let me know. We can send it. Okay. So
is it the James Glazier YouTube channel?
I think so. Yes. But probably, I'm sure Alan has given plenty of talks that have been recorded, but most of them are probably longer than the one we have. Here, while Kali is setting up, I'll find the link for the YouTube. I'll put it in the chat. How's that?
Okay.
Okay. If there are no more comments, we can move on. Kali, I guess you're the last person. Well, we have Kali and then Josh. So Kali, are you ready?
Well, I think I'm about as ready as I'm going to get, at least.
That's
So
okay.
let's-
These can be works in progress. That's fine. And is it okay to record you?
Yes. So for my final project, I looked at a vaccine model for COVID-19. So introducing a vaccination class into an epidemiological model and primarily examining how vaccine coverage rates and vaccine efficacies impact how the pandemic either continues or ends. So first, because I did an epidemiological model, I didn't have as much nitty-gritty biological systems to go through. But here's a bit about COVID-19. So the disease COVID-19 is caused by the SARS-CoV-2 virus, which is a novel coronavirus. The first known case was reported in November. Well, the first case of an unknown illness, which we then identified as the novel coronavirus, was on November 17th, 2019 in Wuhan, China, in the Hubei province. COVID is primarily spread via airborne and droplet transmission. Airborne transmission is breathing in the virus when you breathe the air, and droplet transmission is coming into contact with respiratory droplets like from a sneeze or a cough. Oh, and another thing to mention here, that COVID can be transmitted person to person. Some symptoms of COVID include fever and chills, cough, shortness of breath, and loss of sense of taste and smell. The loss of sense of taste and smell seems to be a rarer symptom, but it is pretty unique to COVID in identifying it from other potential illnesses. And because and I mentioned the symptoms because symptomatic versus asymptomatic cases are going to be important in the models that I use. A symptomatic case is a case where a person is infected with the SARS-CoV-2 virus and symptoms like the ones listed above are displayed. An asymptomatic case is a case where someone is infected with the virus, but no symptoms are being displayed. So the time from the initial infection to the onset of symptoms is called the incubation period, and it's estimated that the incubation period for COVID-19 is around 4 to 5 days. There's something in the chat. Oh, that's the link to the Alan Perelson talk. And in general, cases are asymptomatic during the incubation period. So during the incubation period, you'll be asymptomatic, and then once the onset of symptoms occurred, you will be symptomatic. Of course, there are some cases where people could remain asymptomatic throughout the entire duration of the illness, but for the sake of this model, we're going to be looking at where the asymptomatic to symptomatic switch is going to be important. So papers and sources. So most of my clinical information about COVID-19 came from the CDC. The data that I used is from the Johns Hopkins University Center for Systems Science and Engineering 2019 Novel Coronavirus Visual Dashboard. I provide a link to that GitHub page in my Jupyter Notebook if you'd like to go look at that. The main paper that I looked at was about an SVEIR model for an imperfect anti-SARS vaccine. I thought this paper would be relevant because the SARS-CoV-2 virus is within the SARS family, and we're assuming an imperfect vaccine, and an SVEIR model does account for that symptomatic asymptomatic switch. Some other papers that I looked at were a deterministic time-delayed SVIRS epidemic model. This was a more math-focused paper and less of a practical paper. And modeling the role of asymptomatics in infection spread with SARS-CoV-2, mostly for qualitative reasons, the main finding being that there are a lot of asymptomatic cases and they probably outnumber the symptomatic ones. So the input variables are pretty obvious here. We have the initial total population, susceptible population, infected population, recovered population, and deaths. And I've obtained data for the confirmed cases, recovered cases, and deaths from the Johns Hopkins dashboard. Of course, this is only cases that are reported, so that's a little asterisk there. The output variables being the final population levels for each of the above categories, including where appropriate, where I implemented the vaccinated population. We also take a little bit of a look at the R0 value, which is the basic reproduction number, and for one of the questions I want to answer, we also take a look at the RV value, which is the controlled reproduction number, which is like the R0 value, except when vaccination is included. And a couple of key questions that I wanted to look at in my model were, given a certain vaccine efficacy, how many people would we need to vaccinate? And what's the best method for distributing a finite amount of vaccines? So I looked at 3 different models in this paper, all based off of that SVEIR model from the Gummel et al. Paper. In each of these, I added a D category for deaths from COVID. So that's just for visualizing the deaths. It doesn't really impact the dynamics of any of the other categories because deaths from the illness and natural deaths were already made distinct in that paper. So the first is the SVIRD model. This is where we don't have that asymptomatic class. That's the E class. So susceptible individuals can be vaccinated or infected. Vaccinated individuals can also become infected, but at a decreased rate. And then infected individuals can recover or die from the illness. The SVEIRD model adds the asymptomatic category. So everyone who gets infected will go through that asymptomatic E category before moving on to the symptomatic I category. And a key assumption here that I noticed with the model that the paper made is that asymptomatic individuals do not infect susceptible individuals. So this model assumes that only individuals in the I category can infect susceptible individuals. So that's not necessarily true to real life as well. There's some justification of it is in that if you're asymptomatic, you are less likely to transmit the virus, but of course it's still possible. And finally, I modified the paper model a bit to include an additional S case in which recovered individuals can become susceptible again over time. And natural mortality is present in all of the above categories in every single case. And it's important to mention that, once again, the D in all these models is just deaths from COVID, or what the illness in question. So the measurable input variables are the total population, we know that. And then I got the data for the infected, recovered, and number of deaths from the data. The measurable parameters that you can find through external research are the number of daily births, the natural mortality rate, and the COVID mortality rate from the data. The missing ambiguous information, we don't necessarily know the breakdown of all the infected individuals that we have of the number of asymptomatic versus symptomatic cases, or the rate of becoming resusceptible. I just kind of made up that word. So the rate at which recovered individuals can become susceptible again. So an assumption that I made when modeling, which might not be the correct assumption, probably not the correct assumption, is that all the active cases in the data are symptomatic, which once again is a really big assumption, somewhat justified by the fact that in general, especially in the early stages of the pandemic, people were generally discouraged from being tested unless they had symptoms. So it might be the case that most of the active cases are symptomatic, but again, that's an assumption that I do want to point out. So now we move on to the mathematical model. So this is first the SVIRD arrow diagram. I thought showing the arrow diagram could be helpful, but I could also pull the equations if anyone wants to see that. So we have recruitment into the population, which I just modeled just as births. I didn't account for immigration in this model. We have the transitions between each of these categories. So susceptible individuals can become infected. That's dependent on both the population sizes of S and I, along with the, which I forgot to include here, oops, the effective contact rate is what the paper called it, beta. And of course, susceptible individuals can also become vaccinated at a vaccine coverage rate of Xi. It's so weird to pronounce these letters sometimes. And then vaccinated individuals can also become infected at a rate that's dependent on both the contact rate, but also the vaccine efficacy tau. So the vaccine, so 1 minus the vaccine efficacy is like the chance that the vaccine isn't effective and therefore you can become infected with the virus. So we do 1 minus tau to signify that. Then infected individuals can die from the virus. And then there's also natural mortality in all of these categories as well. Next, the SVEIRD model. So here we're introducing that asymptomatic category in red. So now all infected individuals in the S category will go through the E category before moving on to I at a rate of alpha times E. And alpha is the symptom development rate. So this is 1 over the incubation period. So if your incubation period is 5 days, then your symptom development rate will be 0.2, 1 over 5. And I believe that's the only thing that's necessarily different about this model. And finally, we have the SVEIRSD model. As you can see, I had to get a bit more creative with my arrows. So this is showing how the only addition here is that recovered individuals can become susceptible again at a rate that's dependent on what I'm dubbing the resusceptibility rate, sigma times R. And again, I think that's the only change here. So throughout in my modeling project, I was, as vaccination coverage rate and vaccine efficacy were the 2 primary variables dealing with vaccination. That's mostly what I focused on. Once again, reminding of the key assumptions here, that only symptomatic individuals infect susceptible individuals, only susceptible individuals are vaccinated, and only including births in the population recruitment, not things like immigration or emigration. There were no real alternative hypotheses that I looked at. I did explore a separate SVIRS model, but for the purposes of this Jupyter Notebook, that's kind of more of a toy model. We're focusing more on the other paper instead. And the model was implemented as a compartmental model in antimony. Here's just all of them. So now let's go to the Jupyter Notebook. First bit of the biological background, mathematics, link to the paper here. Explanation of compartmental models, a little explanation on R naught values. Then we go into the SVIRD model. So here is the anemone implementation. And one of the first things I do is just thinking about the educational purpose of the app, examine the model behavior and how it changes based on how each of these individuals' parameters will change. So I just kind of go through them one by one using a slider. The problem with examining the contact rate in particular is that the contact rate has to be pretty small in order to track the differences over time. But the widget slider doesn't like working with too small values. So when we go from a contact rate of 0 to 0.00001, that we see this dramatic jump in cases.
So I was going to make a suggestion both for you and for Tyler, which is that really what matters, the infection rate is really we wrote it, when we did it in class, we wrote it as the probability of getting infected if you meet an infected person times the number of contacts per day.
Right.
And that number is not small. If you scroll back up to the way you wrote it, and Alan does it this way, and a lot of people do it. You write susceptible goes to infected at a rate, contact rate times S times I. But that means that when the population changes, the rate of infection changes, and it shouldn't. So what I would recommend doing is writing exactly the same, it winds up being exactly the same numerically, but it looks different, which is, let me annotate if I can just for a second. Instead of writing it this way, you say sorry. Instead of writing S goes to I at a rate beta times S times I would write it this way. I would write it's a rate, it'll wind up being beta times, in your case, the total population, P total, times the probability that a person I run into is infected, which is I divided by P total, times the number of people available to infect, which is S. So I don't know why S disappeared, S. So in this case, you notice the P total cancels out. But what you have here is, if I run into somebody, what's the probability they're infected? That's the fraction of people infected, the number infected divided by the total population. The number of times that happens is proportional to the number of susceptibles. And the rate of infection per encounter is beta times the total population. So in Tyler's model, it would look like beta times T naught.
Hi, I'm back. Sorry about that. I got kicked off
Okay.
the Zoom call.
Sorry. It would be beta times T naught times I divided by T naught times S. And in that case, if you do that, your beta value will be on the order of 1. It won't be very small. And so effectively, you're not changing anything except that, where's your beta? You see your contact rate, sorry, I lost my annotate. You see your contact rate here is 4.4 e to the minus 7.
Yeah, that's
It's
the value that they gave in the paper.
Right.
Yeah.
It's 4e minus 7, but it really should be 4e minus 7 times the initial population, which is times 1e6. So this is 0.4. Do you see what I'm saying? So I would write it this way. So I would say that the rate, which is we call beta, is equal to whatever they said, contact rate times, you could either use the current total population, which is really better, or the initial one, times S0. And then in your equation here, where is it? It's this one out there. You'd say that this is now going to be written as, instead of saying contact rate times S times I, it would be beta times S times I over S0.
And
Or-
this would occur in every equation where the contact rate takes place.
Yeah,
kind of is
basically,
seen.
anytime you have something that's multiplied by the fraction infected, yes, it would. Because again, the probability that a vaccinated person encounters an infected person depends on I divided by the total population, right?
Right.
I don't care how many people are infected in the universe. I only care what the probability that each person I encounter is infected. And so actually writing it this way, I don't know why people write it that way, actually. It's not mathematically wrong, but it's conceptually wrong. And Josh ran into this when he was working with a group in Pittsburgh, where the units didn't match on either side of the equation because they hadn't thought about that. So I would suggest, again, the simplest thing is just to multiply and divide by S0, which doesn't do anything. The other thing to do would be just to define P total, which is your total population. You have to use a colon equals. P total is equal to S plus V plus I plus R. This is the total number of people in my population.
Right.
And then you would say that you multiply by I divided by P total. And then to correct for beta, your contact rate would just then be initial contact rate times the initial S0. And that gives you beta. And I think then if you pull beta will be a number. Here it's 0.4, the probability per encounter. So what this is saying is that if I run into an infected person, I have a probability of 0.4 of getting infected, which is a more meaningful number than to say that my contact rate is 4 10 to the -7, which doesn't mean anything.
Right. Okay.
So does that I could go over it again, but if that makes sense. And I think Tyler you ran into this clearly in your presentation. And I know I went over this with Brian because we worked on this over the weekend together.
Yeah, I think, yeah.
It clarifies things a lot. And again, I don't know quite why people don't put that into their paper, why they don't do it that way as standard practice. And you don't have to, but I think it'll simplify. Since you mentioned that pulling that slider was a pain, this will make life easier because now your probability of getting infected when you run into somebody is a number between 0 and 1.
That'll make it a lot more intuitive, I think. All right, so we'll keep that in mind. That is a change I should probably make. Well, that I will make, not probably.
Great. It shouldn't change your results very much, but it should make it easier to interpret.
Right. Yeah, so that was the contact rate. Now the recovery rate, the rest of this should be a lot more logical. So for the sake of showing how the trends change over time, we're going to vary the recovery rate between 0 and 1, going up by a step of 0.01. Again, these aren't necessarily intended to be values that would make be logical for the pandemic, but we want to make sure that the model is behaving how it should. So if we start at 0, no one recovers, so we don't have any recovery, and we just see a lot of deaths over time. If we start increasing that, our recovery rate obviously gets increased. And we just keep seeing that increase in recovery rate, which is overall what we're expecting. And as we have more people recover, then we see that the total population also gets higher as well over time. So this is, as we see more people recovering, we're also seeing fewer, well, I guess it's not dependent on, the deaths aren't dependent on the recovery, but we are seeing an increase in recovered individuals, which is what we're expecting if we increase the recovery rate. All right, so now the natural mortality rate, sorry, this is all kind of boring, it's just, I put this into the notebook. So this, I thought it would be best to show the graph separately. As we increase the natural mortality rate, you can see it's kind of subtle that this line here in the total number of susceptible people over time will go down. And then you can see number of recovered people over time will also decrease at a faster rate. And then the total population over time, if I scroll, will also keep decreasing. Sorry, at a faster rate. And then disease mortality. So if we have no disease mortality, then we have no deaths from the disease. And if we increase it, sorry about that, we start to see deaths from the disease. So now I believe that's all the non-vaccination parameters. Dr. Glazier, were you going to say something?
I was just going to say that for some of those parameters where the values are small, you might display the value times 1,000 or the value times 1,000,000. Just the way, if you say to go to Worldometer, they cite the number of deaths per million people.
Right.
Or they cite the number of deaths per 1,000 people. Mm-hmm. So you could always rescale your parameters so that the slider shows the value times 1,000 or the value divided by 1,000, so that the value you're manipulating is a number that's displayable. That's something that applies to everybody.
Okay.
Just so that you can see what the number you're pulling is.
Right. Yeah, sometimes when you change the parameters, if they get too extreme, then you don't really see very many changes over time. All right, so then we look at the vaccination. Is that the vaccine efficacy is a lot more important in determining the model behavior than the vaccination coverage rate. So if we just have a no vaccine scenario. And so, okay, I'm still on. Then we have no vaccinations. We have everything's kind of plateaued by the 1,000th day. I should probably change it to be less than 1,000, but there's probably a reason I did it. If we increase, let's have a little bit of efficacy in our vaccine. If we increase the vaccination coverage rate, we start to see, right, so there's a little bit of impact at the beginning going from no vaccine to a little bit of vaccine, but overall, if we change the vaccination coverage rate without changing the efficacy, we're not seeing very much change in the model. But if we keep increasing the vaccine efficacy, then everything starts to shift rightward and also decrease in value. It appears to decrease in value. So we're seeing number of vaccinations increasing. We're seeing the infection rate's already pretty low, so we're not seeing as much of an impact there. But what the vaccine, it looks like it's doing is prolonging the amount of time until we see infections. And also it looks like, decreasing the number of infections, decreasing that peak there, which is what we want when we introduce a vaccine.
So I'm trying to understand. So you're saying the vaccination rate, what is V? I see the number of V's is going down because vaccinated people are becoming infected.
Right.
Okay. So you're assuming people get vaccinated immediately or very rapidly up to some fraction. Okay, I see.
Yes. Yeah, so there's a certain percentage of the susceptible population that gets vaccinated every day, but see, the susceptible populations decreases so quickly that really that probably explains why the vaccination coverage rate isn't having that much of an impact if we're just decreasing the number of susceptible individuals that fast. So that means the vaccine efficacy is going to be really what's determining the number of infected individuals that we're going to have. So that has been the SVIRD model. Now we're going to the SVEIRD model. Here it is. And as before, we're looking at the model behavior. I don't know if y'all wanted me to go through this again.
You don't have to go through every
Okay,
example, just the ones you think are most important.
Okay, so let's just look at the symptom development rate because that's a new variable. So let's start this at 0. Let's see, waiting for this too. I'm not sure if this is not loading or if there's just no impact. There should be an impact. Oh, here we go. Okay, there we go. So if we put the symptomatic development rate to 0 and we wait, then that means that no one goes from the asymptomatic stage to the I infected stage. So really this graph isn't super interesting. But if we start to increase it, then we're seeing that we have some asymptomatically infected individuals followed in the peak soon after. We're seeing a bit of infected individuals, and we expect that the peak for asymptomatic individuals comes before the peak for symptomatic individuals because everyone has to go through the asymptomatic phase first before becoming symptomatic. Yeah, so as we increase the rate of symptom development, the time scale gets pushed to the left, so we're seeing people becoming infected sooner. And then at a certain point, we'll see the number of symptomatic infections overtaking the asymptomatic ones as far as peak height goes, though the peak for symptomatic individuals always occurs after the peak for asymptomatic individuals. Right, and everything else should be as before. So now we sweep for the vaccine variables again. Start with no vaccination first, wait for that to load a bit. And that might be it. Oh, probably simulate. There we go. So again, the big impact of the vaccine coverage rate occurs going from 0 coverage to some coverage. But again, it's not going to really have much of an impact for the rest of the model. The efficacy, as we increase that, just as before, we see that, oh, sorry, can I take a break for a second? I have a little bit of a leg cramp and it's affecting my ability to give this presentation.
Oh, I'm sorry. Be safe. Josh, do you want to mention what you just said here about how to update the?
So Because it seems like everyone is experiencing the same problem with slow updates as you change the values of the widgets. Whatever you define the widget here, for example, in the interact, the widget is defined inside the interact function. You don't need to, but even if you do it inside the interact function, where Kali typed float slider parenthesis, and after the value you can type comma continuous underscore update equals false, and then you have to do that for every slider. So you do it because that's a property of every widget. So for every widget you have to specify whether that widget updates the updates continuously or not. Depending on the complexity, here's just 2, but in Tyler's case, where he has multiple of them, I would actually recommend defining the sliders as independent variables and then including this in the definition of the sliders.
All right, I'm
Was
back.
that okay?
Sorry.
Okay. You okay?
Yeah, I'm fine. It was just bad timing for
No, I'm sorry. Do whatever you need to do. If you need to take a break, that's fine.
No, it's fine. I'm back now. It's all right. Right, so where were we? We were increasing the vaccine efficacy. And once again, as we increase the vaccine efficacy, we're having everything kind of being postponed due to the vaccine. I believe we're also seeing the number of infected individuals decreasing. Yeah, that peak is decreasing over time and flattening out, which is what we want to see. So now we get to the point where I try to replicate the paper results. Well, I shouldn't say try, I do manage to replicate the results. So just for a little bit of context, I still have yet to include the image in the Jupyter Notebook, but I can show you those images in the paper. So this is the first figure that we're going to try to replicate. Basically, it's an SVEIR model. So they don't have the death category, but that doesn't really impact this result. They give some parameters here. This is with no vaccination, so our vaccine coverage rate is going to be 0. And then the rest of the parameters are as follows in the table that they give up here. That's what we have here. And when we simulate for 300 days, we end up replicating that result. Oh, it's important to note here, this curve for infected individuals, that includes both symptomatically and asymptomatically infected individuals. So that's the first result, probably a good indicator that the model was implemented correctly. And then I also replicate the second result, where this is introducing a vaccine program. We're going to have a vaccination coverage rate of 1/7, vaccine efficacy of 80%. And we're also changing the contact rate to be a bit lower than before. And once again, I was able to replicate that result. So now moving on to the final model, SVEIRSD. So here's where we have resusceptibility. There's got to be a better name for that. So this time I don't walk through every single parameter. I only look at the resusceptible rate because that's the only thing that's different. So as we increase the resusceptible rate, we're going to see a decrease in the number of recovered people over time. And we should also be seeing an increase in, sorry, it's loading. Slight increase in the number of susceptible individuals, but then again, there are so many susceptible individuals that we might not necessarily see that impact. It is interesting that we're seeing a little bit of oscillating behavior here. Yeah, so susceptible is a little increasing, just the number of recovered people is decreasing, which is what we'd expect as people become susceptible again. We're also seeing the number of deaths from the illness increase, which makes sense because if people become susceptible again, they can get infected again and therefore die from the illness. So that is that for model behavior. I promise that's the last model behavior that we're going to go through. And then once again, we sweep for vaccine-related variables. And as we increase the vaccine efficacy, we prolong the outbreak. We're also seeing slight decreases in the number of infected individuals and flattening of those curves, which is what we want for a vaccine scenario. All right, so now going on to the fun part where we actually try to answer some questions. So we have the two questions that we're trying to answer. This is a parameter fitting portion. And I have already downloaded the data for cumulative cases, deaths, and recovered cases, January 22nd through November 28th. And I have the code to extract that data from the files in the appendix, but we're not going to go through that. When we end up plotting that data, we have the cumulative cases increasing kind of exponentially, the cumulative deaths linearly up here, mostly, except for this rapid increase around day 70-ish. Recovered cases also increasing in the same pattern as the confirmed cases. I ended up approximating the number of active cases by subtracting the cumulative deaths and recovered cases from the confirmed cases. And interesting that it also has a very similar shape to the confirmed cases. So notice here we don't really have a true downturn in cases, which made fitting this model interesting. So I chose to focus on days 289 through 312. And I did this because it looks pretty linear here. And I figured that if we were going to introduce a vaccine, the conditions in the later portion of the pandemic would most closely mirror what the conditions of a vaccine scenario would look like. So here's just a zoom in on days 289 through 312 for active cases, deaths, and recovered cases. So I make the assumption that all active cases are symptomatic. And I calculated a rough COVID mortality rate by looking at the increase in deaths by finding the differences between the cumulative deaths and dividing by the number of active cases on that day, because the way we model it, we have I going to D, and we end up with a mean COVID mortality of 0.00019. So that's the estimate there. I do something similar for the recovery rate because recovery is also dependent on the number of symptomatically infected cases. So finding the difference in those and then dividing by the number of active cases to get this. So we have a recovery rate, it looks like a 0.008. So that might seem a bit smaller than what we'd expect for recovery. Dr. Glazier, you look like you're going to say something.
These are daily rates. So I was trying to, so if you assume that the typical person is infected for, say, 14 days, the total death probability from the disease is on the order of 0.003 or 0.004, which is about what you expect, right? The death rate, the raw death rate for COVID is on the order of 0.3%, give or take. So these numbers look reasonable.
Okay.
I was looking because I was just doing the mental arithmetic to try to calculate the number in a form I was more familiar with. But what you've got there looks.
Okay, thank you. So now here comes the tricky part where I try to estimate the number of asymptomatic cases, assuming that all the active cases are symptomatic. So I divided the daily increase in active cases and accounted for the fact that the change in active cases is also influenced by recovery and death. So try to add those back in. And then, add those back into what the number of active cases would be, and then divide that by what I estimated the symptom development rate to be based on a 5-day incubation period by 0.2. And we end up with this graph for the number of asymptomatic cases over time. Not entirely sure what this means. I think it's just, this is a very, very, very rough estimation of the number of asymptomatic cases. But what I really cared about, or for the simulation, what the most important thing to me was, what's that very first value? What's the initial value of asymptomatic cases? And it's actually looking lower than what we expect, because the one of the papers I read about estimating the number of asymptomatic cases in a COVID pandemic came to the conclusion that we should have many, many more asymptomatic cases than symptomatic cases. So this was an interesting result to see that it was less, like a lot less than our number of symptomatic cases. But since we don't really know the breakdown between symptomatic and asymptomatic cases, that to me seemed like what I had to go with for the sake of estimation. But I account for this in my simulation code, where I do allow for a bigger range of value exploration for the initial number of asymptomatic cases. So we can find our starting values for the active, recovered, and deaths from the data. That's pretty straightforward. Here's where we define the optimizer. We're looking to minimize the difference between the model and the data. And then here's where I define the COVID model. And we're going to fit for the contact rate, the symptom development rate, the disease mortality, the recovery rate, and the susceptible rate, because I figured that the number of births, given the sheer number of susceptible people, probably wouldn't matter that much. And we also kind of know that is a ground truth along with the natural mortality rate. So if we do our initial fitting, doesn't look too good so far. Let's see what happens after we end up fitting the whole thing. It's definitely better, but not super close to the data, which kind of might lead me to a bit of the conclusion that maybe we need a different type of model to look at this problem. But continuing with what we've got, here are the new fitted rates. Some of them aren't very different from the expected values. The initial value for asymptomatic cases grew a lot from its initial value, and that's because I allowed for a bit of exploration there, because I knew that we probably wanted it to be a big number. So with that, here is our model that is in theory fitted to the data in the US. So now we're going to look at our first question, which is how many people need to be vaccinated. So in order to do this, we use the condition that the epidemic will end if our R0 value is less than 1. But here, the authors of the Gumel et al. Paper defined a control reproduction number, RV, which is a modified version of R0 to take into account the addition of a vaccination. I don't know if everyone can see that equation, but for our R0 value, we're going to have the number of cases that each person sorry, how do I want to put this? The number of people that each individual infected person will infect in the R0 value is going to be dependent on the symptom development rate and our contact rate beta. And that makes sense because both of those values determine how many people end up getting into that infected category. And then our denominator is going to be a lot of the involve a lot of the death parameters, because those people are then removed from being infected into consideration. So we modify this a bit by including this extra term in the numerator, which is the natural mortality rate plus 1 minus the vaccine efficacy times the vaccination coverage rate. So the 1 minus this term here, the 1 minus the vaccination efficacy times the coverage rate, that will determine how many vaccinated people get infected. So that is definitely important to include there. And then I believe, so here we also account for, we include the vaccine coverage rate along in the denominator with the natural mortality rate. I believe this is accounting for deaths of vaccinated people. So that's our adjusted RV. And we want to look for where RV is less than 1. So what I did here is I wrote a little function that varies RV based on inputs of vaccine efficacy and the coverage rate. And then if we calculate the RV, if it's less than 1, we're going to print that the epidemic ended along with our 2 parameters. And if not, we're going to say that the epidemic is ongoing. What I ended up finding is that you're going to need at least a 100% efficacy or very, very close to 100% efficacy and need at least 20% of the population to be vaccinated. You'll notice if I decrease this to 90%, let's run this again. What? This is not what was happening before. Okay, I promise this is not what was happening before. What?
That's okay. Don't. You've presented a lot, so don't sweat it if something doesn't work.
Yeah, I think the reason was that I had used this model name previously, so I updated it. And now we see that if we only have a 10% vaccine efficacy, the epidemic is still going to be ongoing. So if we increase the efficacy, not really seeing much happen. And if I increase the coverage rate, not much is going to happen either. And even if I go up to the point of 8.9% vaccine efficacy, we still see that the epidemic is ongoing. And again, I don't really consider intermediate values. So like 0.95, we didn't look at that. That could be probably something worth looking into. But as soon as I hit a 100% efficacy, then if we have a vaccination coverage rate of 20%, the epidemic will end. So according to this RV calculation. So that is my first major result. Well, I guess second major result if we include the fact that maybe we should use a different model for this. The second question that I looked into was if there is a limited amount of vaccines, what is the optimal allocation? So I came up with there were 2 methods to model the distribution of the vaccine. The first is continuously distributing the vaccine over a certain period of time. So vaccinating like the same number of people every day. And the second is distributing the vaccine in bursts. So we have a burst of vaccinations followed by no vaccinations, followed by another burst, and so on, depending on how many bursts of the vaccine you have. These functions need to be edited a bit, and I have a few ideas for that. So for the continuous vaccine distribution, we can calculate the vaccine coverage as we have our vaccines per day is equal to the total number of vaccines divided by the total number of days that we define for our vaccination period. So then the coverage rate should be the vaccines per day divided by the susceptible population, because that's going to be the proportion of the susceptible population that we're going to want to vaccinate. The problem with doing it this way is that in the model, our S to V transition is a vaccination coverage rate multiplied by S. So you could end up with a scenario that if your vaccines per day are large enough and this number of susceptible population is low enough that you could end up with a negative susceptible population. So that needs to be workshopped a bit, which I need to put a little more time into that. So now the burst case is that if we distribute the vaccine in bursts, we have to figure out the length of time of a burst, the number of bursts, and then based on how long we're simulating for, the amount of time between bursts. So then our vaccines per burst would be the total vaccines divided by the number of bursts. The vaccines per burst a day are the vaccines per burst divided by the number of days in the burst. So then the coverage rate when we're in a burst should be the vaccines per burst day divided by S, but then we run into the same problem as before. So then on non-burst days, the vaccine coverage rate would be 0.
Well, it shouldn't go negative. S shouldn't go negative because the rate, you're multiplying the rate by S. So no matter what the rate is, if it's a positive number, the vaccination rate times S goes to 0 and S goes to 0. So that should keep your S positive.
But then if we calculate, maybe I need to come up with a new way to calculate the coverage rate, because if the vaccine coverage rate is the vaccines per day divided by S, then the dividing by S and multiplying by S
yeah,
will
then you
cancel.
have to be, yeah.
Yeah, so that's probably what I need to workshop.
So, yeah, the vaccine coverage rate. I mean, that was something I actually was interested. Just to back of the envelope number. If you wanted to vaccinate everyone in the United States with a new vaccine, and you did it reasonably effectively, you'd have to have 40,000 people working full time to do it in a year. So that's actually not as many people as it sounds like. It's 1,000 people per state. That means you put one person in each CVS, and you could vaccinate everybody in the United States in a year. Now you might want to vaccinate faster than that, but it's not crazy. The workload to vaccinate everybody in the U.S. twice, because the vaccine has to be given twice for COVID, is not crazy. If they're efficient. If they're not efficient,
I haven't seen that. So.
Well, they have. I mean, there's just.
I wonder what defines efficiency.
I assumed it took 10 minutes per patient, and they work 10 hours a day, 7 days a week. So, no.
All right, I'll think about that.
But I mean, if you could imagine, it would take 1 million people to do it. It doesn't. It takes 40,000, which is quite a few, but not.
Right.
And they could give multiple vaccinations at the same time. They could give you flu vaccine plus. I think that's a great question. The one thing, I don't want to stop you. I don't know how much more you have to present. So, because you've gone through a lot. What other examples do you want to do? I don't want to stop.
So then in theory, I just adjusted the model by including a total number of vaccines, a number of days that we're going to simulate, then calculating the vaccine per day, and then using that to get the vaccination coverage rate. But as you can see here, when we try to run this, we have a lot of infections. So something needs to be reworked there, and I have a couple ideas for it. Same thing happening with the burst vaccine. We're also seeing a lot of, here the R is the symptomatic infections. Interesting that they lead to the same number of infections. So the jury's out on whether one method is necessarily better than the other. And here's my appendix, and that is the Jupyter Notebook as it stands. So going back. Yeah.
So the one question I had scientifically was, you assume that your resistance after you get the disease has a finite lifetime, but your vaccinateds don't go back to being susceptible. And is that from the paper you sourced it from? Because I would have thought that the vaccine wouldn't, that your immunity from getting vaccinated wouldn't last longer than your immunity from the disease. So I would have included a loop from your V's back to your S's at the same rate your R's went back to S.
Yeah, that's kind of one thing I was thinking about. I have to take another look back at the paper. Is that at the way it currently stands, recovered people can't be vaccinated. So but obviously, like if you're recovered, you could, like in the real world, you could be vaccinated. But I'd have to take another look to see if the paper does anything like that with recovered people getting vaccinated or becoming susceptible again and getting vaccinated.
Becoming susceptible again. I think that's the big concern at the moment. Well, there are many concerns, but one big concern at the moment is if the vaccine only lasts 6 months or 9 months, then you have to revaccinate. With flu, the vaccine lasts 3 months, 6 months.
Yeah.
It doesn't have a very long lifetime. And so, in fact, it's short enough that you should probably be vaccinated in August and then again in January.
Wow.
Which is not CDC policy. But if you talk to people in the business, the doctors get vaccinated that way, even though they don't advise you to.
Right.
But that's fascinating. That's a very comprehensive exploration. Other people, comments, questions, suggestions?
I just had like 2 more slides. Sorry.
Okay. No, I'm sorry. I thought, okay, I didn't know you have even more. Go, please, go ahead. I don't mean to stop you.
Oh. Start from current slide. Okay, so I kind of already talked about this a bit, but it looks like to me when I was looking at the fitting that the model I used might not be the best way to model the situation, at least right now, because we haven't really seen a downturn in cases yet. Could maybe be because of the assumption that all active cases are symptomatic, and maybe perhaps also the inability for asymptomatic cases to infect susceptible people decreases the total number of active cases we could possibly have. Okay. Now, second result, I already talked about, where we would need a very high efficacy with at least a 20% coverage rate in order to end the pandemic. And as we just saw, the vaccination allocation method functions need to be reworked. As it stands now, the continuous method and the burst method lead to the same number of infected individuals, which I thought was interesting. And then if I were to continue this project, the next step would be to look at how long policies like wearing masks and social distancing should be maintained. And this would be, in my vision of it, primarily influenced by figuring out how that probability of infection would change based on how prevention measures were removed. And then the other idea I had was looking into a seasonal model of COVID-19. So then you would have a seasonal vaccination strategy, which could potentially be modeled as a single burst during whatever this COVID-19 season would be. And that is the end of the presentation, I promise. Thank you all for sticking around for that.
No, that's great. That was really impressive. Everybody today was very impressive, especially a week early, so I appreciate it. Comments, suggestions? Again, I think if you write the beta in the way we did it in class at the very beginning, which is the probability, the number of encounters per day of an uninfected person, the probability that the people you encounter are infected, which is I over P tote, and then times the probability that when you encounter an infected person, you get infected. If you break down that beta that way, then it's easier to understand the effect of things like wearing a mask, which reduces the infectivity per encounter, staying home, which reduces the number of encounters,
right,
and so on. But I think so I'm not quite sure I understand that RV, but we could talk about that offline. Because to me, the result is very counterintuitive. I would think that fundamentally, if you vaccinate a fraction X of the population and the efficacy of the vaccine is Y, X times Y is the number that matters, and it should basically wash out. If I have an ineffective vaccine, I should have to vaccinate all the population.
Exactly.
If I have a very effective vaccine, I should only have to vaccinate a small fraction of the population. But it should be the product of those two that matters. And so I have to think about that more.
Yeah. The RV equation I used.
No.
I'm sorry.
But I don't think I'll be able to think. I'll have to think about that one offline, because that I'm not sure I'm going to be able to do that. Other comments, suggestions? I hope the 3 talks today inspired people for next week. Other comments? Please do write up your comments and send them in, and I'll pass them on. Josh, do you want to share your slides quickly?
Yeah, I can do that in 10 minutes.
Okay.
Because there's stuff that I want to point out for people. I know I talked to Tyler about it. I'm sorry. And yeah, my Tellurium just crashed. 1 second. So I guess what I want to point out is, when you are constructing your your application, you have to make sure that that the flow of the application makes sense, in the sense that if, for example, a action below requires the user to do something earlier, you should disable those widgets until the user does what they are required to do, and I'll show it here. So this is the application. You guys seen this. The idea here is that this is just going to be like an educational tool to model the COVID pandemic using an SIR model. And the idea here is that looking at the data, the upside, the downside of the curve, and the upside of the curve, we can estimate the values of the parameters and use those estimates to simulate an initial guess of the model, and then take that initial guess of the model and use an optimizer to get a better estimate of the parameters. So we have the flow diagram here, the equations and the explanation for each of the parameters. There's a lot of text. I'll send this code around and you guys play with it. If I'm still undecided as to the amount of tests is good or bad, but I try to be as thorough with my explanations as possible. Then I provide some instructions about how to use how to use the application. The first part, user needs to load the active cases data. Then they have to estimate mu by zooming in the downside of the curve of the active cases. Then they have to estimate n times gamma by zooming into the upside of the active curve. Once those two have been estimated, then we can estimate R0 and S0. And then once all of the parameters have been estimated, then we can simulate the model with these estimated parameters to get an initial sense of how far we are away from the experimental data, and then we can fit the model. So here the user can choose from the drop-down menu what data set they want to fit. And then the first step is to calculate mu. And then what I mean is you need mu to calculate beta. You cannot, or n times gamma. You cannot calculate n times gamma until you calculate mu. So the button to calculate beta is disabled until the user estimates mu. So for example, we have to look at the downside of the curve. When the user clicks on calculate mu, the software, the program runs a linear fit on that's the red line. So you get an idea of how good your approximation is. And then now the user can calculate beta. If the user changes the range, then the user needs to recalculate mu before continuing. But this is an example of, so the idea is that in this case, you have not a not so good approximation because you can see the linear fit and the data. So this is just, and I've been thinking a little bit about this flow in terms of, okay, when like a subsequent part of the application becomes available. But yeah, once they have calculated beta, and then, so this square here is basically telling you where are you at in the data showed up down here. Then to calculate beta, they have to look at the upside. They can calculate beta, and then now they can calculate R0 from this. R0 and S0, I should add that here. So they get an original estimate of what R0 is. It's 7.09 here. And once they have that, all of the parameters of the model estimated, maybe I can add here like okay, a list of all of the estimations, so it makes clear that all of the parameters have been estimated. then we can simulate the model with these initial guesses. And then after that, then they can fit the model. And then once the model fits to the data, then prints out the estimates and then compares them to the initial guesses. But again, what I guess what I wanted to point out with showing the application here is that in the case that your application requires certain steps to be completed in a specific order, you might need to think about how to enable and disable the options so that the user is forced to complete them in the specific order that you require. So I know Tyler, for example, he needed the user to run first the estimate before running the fitting algorithm. So we worked on making one like as dependent on the other. And that is explained in the widgets in the documentation.
Have you shared this notebook with the class?
I shared a version that doesn't have all. I shared a version on Monday that doesn't have all these flow constraints. That's something I worked on today. But I'll share, like, when the class finishes, I'll share this one with everyone in class. The code can be complicated because it requires you to think a little bit of the flow. And here there is a specific flow in this app. In your apps, there might or might not be such a tight flow in terms of what you need the user to do. But it's always good to think, okay, how is this part of the code related to this other part of the code? And then if they are dependent, then you have to force that dependent. You have to establish a dependency in the code. So yeah, here is fits the model and then estimates. So let's say now the user wants to try a different data set, goes to Spain, then it updates the plots here, it resets them, and it resets the button, so the user has to recalculate all of this. And the last thing I was actually looking at was I want to eliminate the print outputs from the cells below, so these things disappear once you set if I've changed, because this corresponds to the previous data set. So if I change the data set, I want these ones to reset. But I was playing with that one. That's something I'll probably have by the end of the week. But again, yeah, that's another example of how the flow of the application, you have to think about how the flow of the application works. And then try it out, like try out the different options. What happens if the user does this? What happens if the user does that? It's supposed to run smoothly. I'll share this code with you guys. I guess the most important thing is I would say the continuous update. It's important, so you guys don't have to wait for the models to simulate all the time. And also thinking about now that you have developed your notebook, put yourself on the shoes of the user and think as if you were going to this application for the first time, what the application needs, so that it's self-explanatory, that doesn't lose the user if you have, I mean, it depends. For the class, you might want to have 10 examples, right? But for an application, then you might want to cut a little bit and have 1 or 2 that are important. And then if there's examples that are interesting, then point out that to the user, like either as you'd say as a print image of the interesting results, and then you paste them inside of markdown cell, or you actually give the instruction to the user on how to generate those interesting results. Yeah, and I'll share this with you guys.
Okay, great. Are there any other comments, questions? In terms of uploading things, once people think that it's done, we'll talk to Giuliano and Josh, and they'll work on helping you get them uploaded. But it's worth spending some time to get these right, because if they're good, people will use them and your name will be on them. So it's a publication.
Right. You can put this in your CVs because it gets a DOI, like a reference number. So make sure that this is something that you're proud of and that you want to show. Let's say if you want to go to grad school, to your the schools you're applying to, or the programs you're applying to,
or to a potential employer.
Right.
As I say, 2 years ago, we had a student who got a job at Facebook, more or less based on what they did in their little class project. I can't promise that your results may vary, but it happens. So think of this as something that could be something that presents you to the world. It doesn't have to be, but it can be. These do get noticed. Okay, I guess people are probably tired. Thank you, everyone. For next week, I don't like to cut people off. I like people to talk as long as they want to. But since we have 4 presentations next week, if people can try to keep their presentations a little concise, take as much time as you need. But I think maybe having seen what we talked about this week, maybe that will, I hope that's helpful to people for preparing for next week. And again, I'm happy to talk to people tomorrow, Thursday, Friday, Saturday, Sunday, Monday, both the people who presented today to modify things and work through problems, and the people who are presenting next week to help everyone come up with the best possible presentations, so that when we upload these things, they're things that you're proud of. And I'm confident everybody will be. I'm very impressed by what people have done. Okay, any last questions, comments?
Did we just email you our notes? I didn't see a Canvas assignment, but I just wanted to make sure.
Josh, could you set up a place to upload the slides and the reports and things?
Mm-hmm. Yeah, I'll do that right away.
Good point. I that slipped my mind. So we'll put a place for you to do that. It seems the time has passed so fast. It's hard to believe the semester's over. I thought when I created the assignment at the very beginning, that there was an upload, but I guess I lost it. So thank you for pointing that out. We'll fix that. Anything else we want to go over?